Okay, well, we're coming up to the start point, so I'd just like to welcome people as they're logging into the day 4 of the open source chemical science symposium. We've had some fantastic presentations on all 3 streams, both the workshops, the data, and the publishing streams. And this session here is going to be one on the open source tools for chemistry section. Fragment-based screening has become an essential part of any drug discovery program for identifying leads. However, the biophysical techniques to identify those leads in some cases can be rather expensive to deliver, and one of the key ways of doing this is, of course, using X-ray crystallography. The synchrotron at Diamond provides an invaluable resource, and XChem at Diamond provides a relatively painless way for scientists to access fragment-based screening. Today, Rachel's going to tell us all about that work that's being done there and how you can access it. This is particularly topical because at the moment, the XChem group have actually provided many of the starting points for the Project Moonshot program to identify novel therapeutic agents to treat COVID-19. So I'll now make you the presenter, Rachel. And so it's over to you whenever you're ready to start.
Perfect, thank you, Chris. Okay. So you can see the right slides, you're not seeing the presenter move or anything, are you?
Yeah, that's absolutely fine, and we can hear you perfectly.
Okay, perfect. So just if you want to have a quick look at Fragalysis as we go, the production server address is just up here, so it's fragalysis.diamond.ac.uk. But I'd hold off a bit until we actually get onto the application stuff. So the title that I've put for the talk is Fragalysis, an open source web platform in the cloud for reviewing and progressing fragment hits from the beamline. I'll talk a bit more about the science behind what we do and how we do it as well before I dive into Fragalysis. So Fragalysis is developed across 2 organizations, so across Diamond and the SGC, and also involves a lot of contributors across the world as well. So we have separate developers in Slovakia for the front end stuff. We work with some informatics companies as well. And we work very, very closely with Janssen Pharmaceuticals. So they've actually funded a lot of this work. So that's just a sort of upfront acknowledgement of that. So just a quick outline, I'll introduce what fragment-based drug discovery is, because I know that, chemistry is quite a broad area, so I'll just cover that quickly. And I'll go into a bit more detail about our experiment at XChem. This might be a bit controversial given one of the earlier talks was on PyMol, but I'll try and explain why we think for this particular experiment, something like PyMol for visualization isn't usually the best solution, and how we can think about the problem in a different way. And then I'll introduce Fragalysis as a tool in the cloud and go through how it's used and some of the data that's on there. And if we have time at the end, I'll go through a live demo of installing our Fragalysis API and interacting with that to get access to some of the data in a slightly different way. So Diamond, we're about 16 miles away by bike from Oxford in the UK. I'm sure everyone has heard of Oxford, but not necessarily Harwell. And so we're the UK's national synchrotron at Diamond. So it's a light source, a particle accelerator. And we have a beamline there, which is an experimental station, and we're based at a beamline called IO41. So at Diamond, in their life science offering, there's a big macromolecular crystallography village. So there are 4 operational beamlines, so that's standard access to experiments, and some experiment-specific beamlines as well that offer different variations on standard X-rays for different types of experiments. So things like long wavelengths and in situ screening, room temperature experiments, that sort of stuff. And there's a lot of other stuff that you can do with particles when they're accelerated as well. So there's a lot of stuff in the physical sciences and outside of macromolecular crystallography as well. There's a new facility for cryo-EM too, which is exciting. That's called EBIC. And then I suppose obviously the most important one for this talk is XChem, which lives at the IO41 beamline. So these beamlines are experimental stations that come off the main ring. So the particles are accelerated around the ring, and then we use magnets to bend the light off into these experimental stations and use the wavelength of light at a specific point to perform different types of experiment. So also involved in this in Frank's group is the SGC. They've actually changed names now. They used to be called the Structural Genomics Consortium. So they started operations in 2004, and it's mostly funded by government agencies and the Wellcome Trust, with lots of charities and also a lot of buy-in from leading pharmaceutical companies. So the SGC is a massive consortia of more than 300 people, and it runs across 6 different countries. So there's Oxford, Canada, Brazil, the US, Germany, and Stockholm as well. And their main remit is human genes, so things like X-ray structures, drug discovery enabling reagents, and chemical probes as well. One important thing about the SGC is that they have an open science policy. So they want to ensure that results, reagents, and expertise in their area of remit is placed immediately in the public domain when it's ready. And importantly, SGC scientists never file any patents as well. And this is just a group of some of those organizations that are involved in funding and working with the SGC to make this stuff happen. So drug discovery specifically, where do you start? Historically, methods in drug discovery were quite random. So you have things like serendipitous discovery, so that's your penicillin story kind of thing, and things like natural product screening and phenotypic screening and classical pharmacology. And over time, the methods involved and the way we think about drug discovery has become a bit more rational. I wouldn't say we're at a point where we're actually doing rational drug discovery entirely yet, but we're getting there. And a big part of that is library-based screening. So designing sets of molecules that are focused on the problem of designing a drug in a specific way. And I'll go into that in terms of fragment-based drug discovery specifically in a minute. So when you're designing a drug, what you're hoping for is a potent molecule, so something that inhibits your protein well. And the smaller libraries that we tend to use give you guaranteed weak binding, and the idea is that you want to achieve that potency through chemistry, and that's what fragments are. There's a big difference in the different kinds of libraries as well, which I'll come into in a bit more detail now. So in fragment-based drug discovery, we're using small molecules that are generally less than 20 heavy atoms. If you think about your traditional screening libraries in high throughput methods, not necessarily crystallography, you're looking at much bigger molecules that are considered more drug-like. The advantage of these sorts of molecules is that there are much more opportunities chemically for interactions between the molecule and the protein. But just because that's the case doesn't mean that the interactions you make with the drug-like molecules are always favorable. So you might be able to get a drug-like molecule to fit in your protein, but you might have lots of areas that you're not hitting and wasted chemical features within those molecules. So instead of using this sort of molecule in fragment-based drug discovery, you use these smaller ones. So there are fewer interaction points available on this sort of molecule, but because of that and the fact that they're smaller and they can get into the protein, they tend to bind a lot more easily, which means you get more hits from this sort of screen than a traditional high throughput sort of screen. And so what you end up with is a lot of hits of very weakly bound molecules. And what you want to do from that point is take the information that you've got and optimize it optimize those molecules with medicinal chemistry. An additional advantage to these sorts of molecules, of course, is that you can uncover structural insights in your protein. So with these sorts of molecules, you might start to hit things like allosteric sites or cryptic binding pockets as well. Okay, so a little bit about structure-based drug design. So structure-based drug design is the idea of using information about the protein itself to inform how you design your drugs moving forward, and not just looking at the ligand and how you can change that. So if you take a group of structures and look at them together, you can start to identify things about the protein like hydrophobic areas, and you can design your drug specifically to fit into your protein. And this is, you know, done in a certain way. So you start with these designs and computational chemists could iterate on them. And then behind that, you need to actually make the compound. So you have medicinal and synthetic chemists working on hundreds of compounds at a time. And on top of that, you need some biophysics to measure how well those molecules actually bind to your protein. And what you end up with an output of this process is hundreds of nearly identical crystal structures, because you're just making small changes to the molecule over time. So it's not a fast process. This slide hasn't turned out well. I apologize. Okay, so this slide is trying to show what happens traditionally. So on the left-hand side here, we've got a fragment library, followed by a binding assay and a structure. So traditionally, in a binding assay, you'd have, you know, a few data points all put into the same protein, and this gives you some limited sensitivity. And in the long term, even with a binding assay, you really want a structure so you can see what's going on between whatever that molecule you're hitting with is and the protein itself. Another terrible slide, I apologize. So when we move to a fragment library in a high throughput screening method, we've got a lot more compounds that we can get into the protein. It's a very sensitive technique. You get the 3D structures out as well as a result of it. And traditionally, you know, you achieve this by using a high compound concentration. So historically, it was really difficult to do this as a high throughput experiment because crystallography takes time. So you could traditionally only do hundreds of crystal structures, and it might have taken you months to get through that. But over time, the technology has really taken off. So with the introduction of things like synchrotrons like Diamond, different detectors, automation of the experiment itself through robots, and also different algorithms, and, you know, the increased power of computers as well. We've sort of gotten away from this problem of the experiment taking too long. And so it then became a job for a facility. So you want to make this sort of screening fast and accessible. And that's where XChem came in. So this is just a quick outline of what we do at XChem. So we have fragment libraries that live in multi-well plates. So each well in this plate will have a different compound in it in solution. And we also crystallize our proteins into plates as well. And essentially what we do then is we decide what compounds we want to put in our crystals once we've identified the crystals and where they are. And we use acoustic drops in a special machine to shoot the compounds at specified points into the protein drop to hit the crystals. Once you've got to this point, there's a method and a machine at Diamond that will help you mount all of those crystals onto pins so you can put them onto the beamline. And at the beamline, we have a robot called Bart who will pick up hooks of pins and mount them onto beamline so that the data collection itself can be very automated. And what you get out of that is a big pile of data of fragments bound to your protein after you've done a lot of data processing. So we're screening directly into crystals with one compound per crystal. All of the things I've just talked about are automated as well. So now instead of hundreds of crystals in a screen over maybe a month, you're getting to 1,000 crystals in a week. And As a facility, XChem's had routine users since about 2016. These are some old numbers. They're much higher now. I think they did about 125,000 crystals in their first 3 years with over 3,000 hits for about 130 targets. I think at this point we've done academic projects around 200 different targets. So this is just to demonstrate what comes out of an XChem screen, really. So this is the NUD5 protein, which is a project one of our students at the SGC is working on. And so she ran a crystallographic screen at the beamline, and what she gets after she solved all that data is lots of these small molecules crystallizing in different areas of the protein. And you can start to see from this sort of image, you know, there's a problem with the output. What do you do with it? It's a bit of a mess. And so the question becomes, well, what do we do with this mess if we're trying to do drug discovery? So we've got a lot of data over here on the left-hand side. And how do we get to what we really want in this process, which is a drug? And the key questions there are, what can we make fast and cheaply? So most of our users are academic groups, they're not pharmaceutical companies. So they don't have the typical kind of funding streams that pharmaceutical companies would have when they're going through this process. Another question is obviously, what should we make? And to think about that question, you need to think about the data that you've got from your screen and how you can use all of the data to its fullest potential. So one way of thinking about this, I suppose, would be to take your protein, apply some amazing algorithm to it, and hopefully get a drug out at the end. But that's a fail. That doesn't work. That doesn't exist. I don't think anyone's managed to do it that way yet. Maybe in the future, but not now. So moving onwards, normal fragment structures, so the things that come out of this experiment, they're useful, but they're not cheap. Developing them is still expensive, so when you're elaborating them, and it's quite bespoke. The best practice for this is not clear, and each target project is sort of handled ad hoc from XChem. So we primarily, for a long time, have offered support in the screening and then collaborated with academics and pharmaceutical companies after that to develop things further. But that's always generally involved a lot of expensive work. And so we saw a general opportunity in this. So XChem as a facility is quantitatively and qualitatively different. So we have a lot of data and it's very high throughput. So for an individual target, you have more data than you would traditionally have if you did a focused fragment screen. The problem is that no one yet knows how to exploit all of that data, and it's also not yet funded correspondingly either. So there's a specific new opportunity to work out what we do with this data and applying those new opportunities to get us closer to, you know, that golden ticket, which is using these fragment screens to make a drug at a facility. And that could include things like artificial intelligence and robotics. So PyMol. So if you were looking at the output of a fragment screen in PyMol, I think this is pretty much what you're looking at. You'd want to overlay all of your different structures, put them together, look at the ligands together. I think it's just quite a complicated picture. Like, how do you navigate that in your mind, never mind physically? And surely there must be methods that would help us to do this in a better way. So a few years ago, one of Frank's old PhD students, who was then a member of staff and now works at Exscientia, Anthony Bradley, thought about this problem for his PhD, and he developed something called WONKA. And the sort of premise behind that was objective, novel, complex analysis for ensembles of protein ligand structures. So when we're talking about ensembles, we're talking about multiple structures of the same protein with different things bound into them, with subtle differences between the proteins and the ligands. And the sort of things that you sort of started to think about analyzing in those ensembles were ligands, residues, and waters. So you can think about things like pharmacophore formation and how ligands align to pharmacophores, and what the differences are between ligands by fragmentation. You can also think about feature clustering, so in terms of the conformation of your protein or different waters, or how ligands interact, how do you cluster that together into something that's quantitatively meaningful? And also, how do you visualize that as well? So this is just a screenshot over here of the original implementation of WONKA, which is also a web page. I'm sure you'll find the old version of it if you Google it. So what he's displaying over here is a pharmacophore, so different types of pharmacophores, like hydrogen bond acceptors and donors. And then across here, each one of these squares represents a ligand. And if it's colored in green, it means it has an interaction with that pharmacophore, or it fulfills that pharmacophore. And if it's in white, it does not. So you can start to see at that point how we were thinking about looking at structures that come out of this experiment in a different way. So just going into a bit more detail of why that works. If you look on the left-hand side, you can see a complete ensemble. So this is one of those representations of multiple crystal structures all together. And you can see that it's messy. And you know, it doesn't matter whether you use PyMol or whether you use some fancy web application to stack those structures together. It's very difficult to look at. And it's extremely difficult to compare different conformational changes as well. For example, in your protein, which are quite important. So you can see sort of areas where things are moving around, but there's no real good way to quantify that, and you'd sort of rely on a scientist to make those observations and write them down traditionally to notice those patterns. So with WONKA, what was implemented was clustering of these conformational changes. So on the right-hand side, you can see for this particular set of residues, they've been clustered into different conformations and coloured. So then you can turn everything else off in the protein that you're not interested in and just focus on the interactions that you are interested in and start to observe more clearly what's changing conformationally and what that might mean in terms of what your ligand is doing to the overall structure, and take advantage of those in your follow-up designs. So additionally, we can think about that in the context of waters. So on the left, again, is an ensemble of protein structures and multiple waters plotted as blue spheres. So you can see areas where they're sort of similar for different structures. I mean, your alignment algorithm might have some effect on that, but in general, it's quite difficult to visualize what's going on with the waters overall. So on the right-hand side, what Anthony implemented in WONKA is clustering of those water positions and identifying within binding sites where the key water positions could be through representation of these clusters as big bulky spheres in the context of the ensemble. And then finally, we've got the idea of clustering pharmacophoric features, which is the thing I explained on the first slide, where you can define all of the pharmacophores in your structure and use them to direct your design based on which pharmacophores are hit by which ligands as well, and very easily look across ligands to see which ones do and don't fulfill certain pharmacophores. So another thing that looking at this data in an ensemble rather than just a big bunch of different protein structures is useful for is for spotting errors and other interesting things. So I guess this is more applicable to things like the actual structure solution and refinement. But if you start to treat the structures as ensembles, you can quantitatively or qualitatively start to spot outliers. And there's a great quote here that just says outliers are either errors or interesting from a crystallographer, which is good. So we can apply this both to crystal structure data and to computational data as well. So I think this is an example from a a simulation using MD to improve crystal structure solution, and you can see differences in conformations and where they lie and how often they happen. You could use that sort of stuff to say, oh, well, is there quite often a conformational change going on over here? And if so, why does it happen? Or is the protein very flexible? Or maybe if there's, you know, a very, very low percentage where it's happened, is there a mistake in the refinement method itself that's being used to solve these structures? So now that I've gone on
time for one quick question?
Yeah, of course, absolutely.
Yeah, looking at the things that you've just described, and one of the questions is, do we need to predefine the interaction sites manually? I presume that means the active site, or is it generated automatically?
For WONKA. I'm not, in Fragalysis, you don't need to predefine any sites or anything. It's done in an automated way, and I'll cover that later. For WONKA, I'm not 100% sure. It's an old piece of software, and it's really just there to demonstrate sort of where we've gone with our thinking over time. I would imagine no, because we don't tend to think. I mean, like, so the purpose of these software is to help a medicinal chemist to move these structures forward. So we don't really think about this data in terms of sites. Of course, the crystallographer and the person that's interested in the biology will know all about where the active site is, but we tend to think of it more in terms of interactions that are going on between the structure and the ligand, and how we can exploit them to develop drugs in terms of the chemistry.
That's great. Thanks very much.
No worries. Okay, so what is Fragalysis and why did we make it? So I've roughly outlined what the process of the experiment is at Diamond, but I'll just go through it in a bit more detail and sort of explain what the problem is at the end as well. So when you come to do an X-ray screen at Diamond, you start with a target protein. So usually if you have a proposal for a project, one of the things you have to show is that you've got a good crystal system and you can crystallize your proteins well. So that's your starting point. So you come in and you crystallize all of those proteins into plates. Then you move on to the soaking stage. So at this stage, you're putting those small fragments from the fragment libraries straight onto your crystals. Then you shoot those crystals at the beamline and collect some data. And finally, you do some data processing. So you use various methods to take the electron density and convert that into structures so that you've got a 3D view of what fragments are binding to your proteins. At Diamond, underneath all of that, we have some automated pipelines that curate all the data and put it all into one central database. And so what comes out at the end of this process is some structural data. And for a long time, that really was the end. Like I said, everything else was ad hoc. But in an ideal world, offering this as a at least chemical probe and lead discovery facility for users, what you really want to come back is follow-up compounds and have them screen again with the facility and build up in rounds of subsequent optimization with their medicinal chemists and computational chemists. So really what Fragalysis was initially designed for was to try and fill that gap between data coming out from a fragment screen and people not really knowing what to do with it, so that we could get our users back and support the academic community in being able to do this sort of stuff more routinely, and to sort of pose at this idea of looking at the data in a different way. So that's just coming back again to what our typical screen looks like. So the overview and goals in the development of Fragalysis. XChem, we identified that XChem needed significant post-processing and analysis to maximize the impact of our user program, to make this stuff easier. And that the academic community probably needed access to all of our data as well. So this is talking about the guys that actually develop the algorithms that help us get better at designing drugs. and probably that they need a home to test their own tools in the open as well. And probably, you know, pharmaceutical companies do lots and lots of target screens, but we probably do more than each individual pharmaceutical company. And I think the problem with traditional drug development is that there's a lot of IP around the data, essentially. So people are very interested in their own data and keeping it private because, of course, they can make money off it in the end. So why would they share it? At Diamond, we take a different approach in that the data are open. So if you come as a user to Diamond and you want to do a fragment screen and you're an academic group, we won't make you pay any money for it. But the deal is after 3 years, you have to make that data open. And so that puts us in a unique position in terms of how much data we actually have. So thinking about all of those things, what we needed was a platform for accessing the derived data for our users and the academic groups. So that structural data that comes out of the experiment, some sort of an interactive web application to provide access to it and some further analysis. And then also eventually, we're not at this point yet, but some flexible high performance computing as well to run and test further tools. So those computational tools that we hope to bring in that will help us get better at drug design. And ultimately what we want to do is capitalize on the novel and the robust infrastructure developments so that we can create stuff that can be reused by other groups as well. So throughout the development of Fragalysis, we were very focused on making sure it was deployable so that any university or other organization, pharmaceutical company could take that piece of software and deploy it for themselves if we weren't able to reach capacity on offering it as a platform to serve data for the world, I suppose. And so thinking about the computational side of things and developing new molecules, I thought it'd be nice to just go through what a typical computational workflow would look like for designing a new molecule after you've got 3D structures of fragment hits. So your first stage is triaging. So once you've got all of your data off the beamline, you need to work out what you want to work on, which hits are important and which ones do you want to make better and develop into drug-like things. And you have to remember at this point that these small molecules have really poor binding affinities, so they don't usually bind well to the protein. So we can't use biophysical assays to decide which ones are the most important ones. So at this point in a fragment-based campaign, you're thinking about filtering to triage these initial hits. So you're looking at either structural or ligand-based methods for filtering and using that to generate hypotheses to improve the potency when you start elaborating on your molecules. So to design new molecules, you need to do something called enumeration. So typically you use libraries of molecules that we know that are accessible synthetically or that you can buy. And when you're searching through those libraries, you need to think about how you want to modify the hits that you've triaged in that first step and how you do that in a way that aligns with the hypotheses that you've generated during your triaging stage. And so finally, you need to make a decision on, from that enumeration, which might give you thousands or tens of thousands of compounds, which ones are you actually going to buy? And so this comes down computationally to scoring. So you need a metric, a quantitative way of evaluating how well those molecules fit your initial hypotheses. I suppose it could also be qualitative, but if you're looking at 10,000 structures, it's a big task. So in terms of compound enumeration, one method that we thought about early on was the idea of poised reactions. So the idea behind a poised reaction is that you start with fragments in your fragment library that you can easily adapt by simple chemical reactions. And so you have a set of other compounds that you can react with your initial hits and build new molecules. And then you can also purchase different versions of the same synthons, with the synthon being the thing you know can have a reaction with your initial hit, and finally make analogs that you can put back into your screen. So this was something that was done back in 2016. It's quite a robust method, so because this chemistry is so simple, you're able to make drug-like products quite easily and quickly. And at that point, we thought it could also be possible using commercially available starting points with a compatibility over a range of substrates. What we've moved on to recently and what's implemented in Fragalysis, which I'll talk a bit more about, is using a fragment network. So a paper came out a while ago, 2017, from Aztecs, where they built a fragment network. And the idea behind this is you separate your molecules out into component pieces, and you work out what the relationships are between those molecules, and you represent that in a graph. So on the nodes of your graph, you have the molecules or the parts of molecules themselves, and then along the edges, you describe what the chemical transformation is to get from one molecule to another, and you encode that as smarts. A few of you might not be familiar with that, but it's just a way of representing a change. And so those changes can be things like adding a group to a molecule. It could be making a replacement to the linker, or changing part of the molecule entirely as well. So I'll just go through very quickly how this works at Diamond and how we get our data into Fragalysis before I show you the goods, so to say. So we have masses of data collected as part of a XChem screen. We scrape all of our data together and put it up into a central database, and we have a continual process looking after that. And then we upload our hits to the cloud, and what we hope is that eventually the tools within the Fragalysis cloud will allow the movement of these initial hits to viable lead compounds. And so the final result coming out of Fragalysis would be that a user selects a number of interesting leads and pulls them back down from the cloud, has a massive new pool of compounds, and comes back to XChem screens and goes through that cycle to optimize over time. So focusing on Fragalysis specifically, this is an old screenshot, but it will make a point. There are features within Fragalysis that demonstrate some of the points that I've just talked through, especially with the graph network stuff, exporting results as well. One thing to note is that everything that's encoded in our graph network, which I promise I will show you, is a molecule that's commercially available. And most of them are available from Enamine, who are a CRO. So if you designed a new molecule using our graph network, you could send off a list of the smile strings of the compounds that you're interested in to Enamine, and they will give you a quote. And typically, I think those molecules cost about £250 each, which is very cheap. There's about a 3 or 4 week turnaround, and I think they have about an 80% success rate on synthesizing those molecules as well. So Fragalysis, I will show you Fragalysis in a second. One thing to point out before we start is that there are sort of 3 main bits of Fragalysis. There's the left-hand side where we're looking at crystallographic data. There's a 3D viewer in the middle, and then on the right-hand side, we're thinking about computational data from computational chemistry experiments, essentially. So on the left-hand side, we're thinking about the target and sites, the protein structures with and without ligands, and looking at things like properties. And then on the right-hand side, we're looking at enumerated molecules that have been converted to 3D structures, and different scores and how we can filter them to help us decide what to buy. Okay, so visualization of fragment data. So,
would this be a good time to take a couple of questions?
Yeah, absolutely.
So one of them's rather more technical for the crystallographers. How do you cryoprotect your crystals in the workflow?
I don't know. You would have to ask the beamline scientists. All of the information is available if you Google fragment screening XChem. Our fragment screening pages have all of the details of exactly how they do the experiments.
Okay, that's great. And you talked about available from Enamine. Enamine has a database of, what is it, 1.3 billion structures. So do you have all of those enumerated?
Something insane like that. No. So our graph network is focused on our fragment libraries. So you can sort of take a subspace of Enamine's real database that aligns directly to your fragment library, so that you know that if there are molecules in the wider space that aren't accessible from the starting points that we have, we can just start to ignore them. But of course, as we have more and more molecules being developed, the size of them gets bigger and the diversity of them gets bigger. So we reprime our graph network every now and again and introduce new molecules so that we can make sure we're covering the elaboration space that's available to us.
So just to be clear, all of your fragments are reagents in the Enamine real database, are they?
A majority of them, yes. So we have a few different fragment libraries for different purposes, but the main one is the DSI Poise library, which was developed in collaboration with Enamine to align to their real database and to make sure that we can elaborate them very easily with their building blocks.
And then another question on this. How do you determine if a reaction between the fragments are easy? Do you incorporate reaction database into the algorithm?
Yes. So the transformations that we can do are based on transformations that Enamine know they can do based on the building blocks that they have available to them and the reactions they have available to them as well.
Okay, I mean, and they claim something, what is it, 80% success rate? So
yeah.
Okay, that's great. Okay, I think that's all the questions for now. So let's head on.
Okay, cool. So I'm going to jump on Fragalysis at this point because I think we're 40 minutes into a talk and I haven't even showed you the platform yet. Okay, so this is the landing page you'll get to when you get to Fragalysis. So that's if you go to fragalysis.diamond.ac.uk, this is the landing page. And what you're presented with over on the left-hand side is a list of different targets. So all of this data is open source data that's come from either our academic users or the SGC, who are arguably our biggest source of data, but that's just because of what Frank's group do. So there's quite a lot of data there. I'll focus on the MPro data because I think people are interested in it, so why not? So if we navigate to one of those target pages.
I've just put the link in the chat, so I don't know if you want to just pause for a few seconds to let people click on it if they want to follow along.
Yeah, absolutely. I'll just give you a second to get there.
Yeah. So the systems can accommodate 80 to 100 people all joining at the same time.
I hope so. I think so.
Yeah. Okay, that should be fun.
Okay, cool. So what you're presented with is the Fragalysis page. So Fragalysis is designed to work as a single page application. So everything you do for a single target will all happen in this page. And so I'm just going to talk through briefly the different sections and how we interact with it and sort of the theory and stuff behind them. And that comes back to the slides a little bit, but actually maybe I don't even need them. So the first thing that you'll see is something called the hit cluster selector up on the top left. And what you're presented with here is a reference protein in the background represented as a ribbon, and then all of these blue spheres over here. And if a sphere is colored in green, it means it's turned on. So if I click on one of them, it should turn off. And if I click on another one, it'll turn on. So you'll also notice that these spheres are of different sizes, and the size is representative of how many fragments are in that site or that area. So when we're generating these sites, when we process and upload our data, there are two methods we can do that by. So there is one way you can do it, which is an automated clustering using something called a DP means algorithm. I won't tell you what that means because it's probably quite boring to a lot of you, but it essentially, you don't define how many sites you want in the first place. It just uses the data to generate a probabilistic model of where they should probably be clustered. So if you had an experiment with a lot of data and you weren't really sure what to do with it or what your sites were, then you might choose to use that algorithm in the first place to start to identify different sites and areas of your protein. The other option we have is that our users can define what the sites are. So for MPro, for example, we have, in this case, the scientists have not decided to name things as active site, but they've chosen to categorize the structures. So you'll see there are a few sites that are the names of different chemical series, for example, and then there are some sites that are things like active site molecules that have come from the moonshot, or active site molecules that came from the XChem screen. And they've also made a point of separating out things like covalent inhibitors as well, and things that crystallize at the dimer interface. Okay, so the next part of the application is the hit navigator. So this is where all of your ligand structures come up. And at this point, it's important to point out that in Fragalysis, we're not thinking anymore about crystals, we're thinking about ligands. So if you had a crystal structure that had a ligand crystallized in multiple positions, before we upload that data to Fragalysis, we separate those ligands out. And in Fragalysis, we're treating the protein and ligand both in isolation from each other, so that we can work on the ligands on their own or together through some of the visualization options. So if I find an interesting site, but it's quite slow, there must be a lot of you on it. Wonderful. Maybe I'm frozen. I'll just try and reload the page. Okay, so maybe it's just about handling it. Okay, so when the page first loaded, I'm sure you will all see there was a ligand turned on. So one of the best things about Fragalysis is that when we're looking at those sites, we can start to overlay molecules together and see what sorts of things are quite common. So you could look at it from a ligand point of view and look at these molecules and say, oh, you know, there's a whole bunch of fragments that have this oxygen atom and the ring systems on either side. Okay, cool, so what's going on? So the next thing we can do is we can start turning proteins on. That's not going to work for some reason. Did that work? It did. You can start turning proteins on, and we can have one or two of them on together or none at all. We can look at them for one ligand at a time. We can also turn on interactions. So if I've got that protein and those interactions on, if I turn off this protein, and so you can look at the interactions for an individual molecule if you want and start to identify what they are. So you can see things like weak hydrogen bonds, normal hydrogen bonds, pi stacking, all sorts of things, both in the immediate area of the ligand and around the protein as well, if you're interested on what's going on further out. And if I messed up my view, quite an interesting feature is that we can choose to center on a ligand. So you can get yourself back to what you were looking at and what you were doing before you messed it up. The other thing we can do is turn on a surface. So this is an electrostatic surface. And so you can use this to sort of look at the shape of your protein and what the electrostatics around the ligand compared to the protein look like. And it's quite useful when you start looking at multiple ligands. If you're, for example, looking at this site here, which is the I believe it's the active site in MPro, you can see how much coverage you're getting on that site as well. So in MPro, there are sort of 4 sub-pockets that each have different functions, and there's also the most important bit, which is this, not that, which I will find later and show you when I go through a specific example of how someone's used this for compound design before. So if I turn this off, we can get everything off. And of course, in the middle here, what we're presented with is the template protein. In terms of visualization in the central viewer, there are also some extra controls you can use. So to start with, if you have been struggling to use that viewer, if you click on the mouse icon underneath the central viewer, you'll get a list of all of the different mouse controls. And I think these are set to work similarly to Qt by default. Some simple controls, so the settings controls, the little cog icon underneath that central viewer will allow you to switch your background color to white, which I prefer, but apparently crystallographers prefer black, so it's black by default. You can change the clipping, so that's how much of the protein you can see from the front, and you can change the clipping from the back as well. You can also change the distance of clipping if you want, and you can add some fogging as well, which will sort of, well, does what it says on the tin, create a fog over the protein so that it fades into the background a bit, and you've got that for both the near and far parts of the protein. Okay, so if we go and turn a ligand or a couple of ligands on as well, and we center in on one, I can demonstrate some of the other features. Just move around a bit. So let's change that fogging because I just made it look awful. Okay. So this central button underneath the 3D viewer is for display control. So this is controlling exactly what's rendered in that 3D viewer and having some control over the different ways to visualize things, similarly as you would in PyMol. So for example, we're using the same labels in the name of the thing, and you're indicating whether you're using a ligand or a protein as well. So if we wanted to change this ligand here, which is the top one, we can use the drop-down menu to see what representations already exist. And then we could, for example, change it to a licorice representation. We can also access some advanced settings, which do various things. So these are essentially all of the settings that are available in the visualization library that we use in Fragalysis. But if you have a play around with it, you can do some quite interesting things. I think obviously one of the easiest things to do is just change the color of the molecule, for example. Yeah, so there's some advanced options available for you there, and you can also control for an individual ligand things like your clipping and your opacity, and make them more translucent. That kind of thing might be more useful when you've got lots of ligands overlaid on top of each other, so you can sort of control how you're looking at this stuff. We can also add an extra representation. So if we wanted to keep our representation underneath and then add maybe something like a surface over the top, we can also do that. And then we can play with the advanced options to change various things. So now I could create a surface where you can see the ligand through it. We could change things like how the radius of that surface is generated. So we're currently using Van der Waals radius. We could use covalent radius if we wanted to. And we can change the type of surface as well, I believe, somewhere. Yeah, so at the minute we're coloring by element, but you might want to look at it an electrostatic surface for some reason. I don't know why that didn't work, but it didn't. Imagine it's a beautifully coloured electrostatic surface, and we'll be all right. You can also keep those representations in those display controls and turn them off as well by using these eye icons, or you can hide the entire representation. So that's quite useful if you want to keep a lot of ligands on because you're looking at a particular site, but you want to very quickly just hide things and turn them back on without losing the settings by turning the ligand off. And we can also delete the different representations we've made as well. So that's quite a useful feature for playing with the central viewer. So what else can we do? I talked about triaging hits.
Do you want to answer a couple of questions now?
Yeah, absolutely.
Yeah, okay. Could Fragalysis be used for larger ligands like glycans, which are large multimer structures?
I believe so. I think the biggest challenge there would be whether the visualization library is up to it. So we use something called NGL Viewer on our back end, which is the same thing that the PDB have used for a long time. But they've also come out with something called Molstar now, which is a similar library for visualization, but it renders bigger molecules better and faster. So I think if you're looking at bigger proteins, that's where your limitation will be. If you're looking at bigger ligands, I think it would handle them fine. The biggest challenge in the ligands themselves is pulling them out of the proteins in the first place. So when we upload the data, we separate the ligand out completely from the bound state PDB file.
Okay, another question. Can you include biological data?
Not yet. I'm sure we'll get there. So the infrastructure's developing rapidly. So the thing I'm about to talk about, we're doing things like including molecular descriptors, for example. I suppose the challenge is including that data in the database that lies behind Fragalysis. What we can do, so one very nice feature of Fragalysis is that you can download structures. So if I download, for example, the MPRO data for only the 17th time since I last emptied my recycle bin, we can provide as part of the download package that we give, if I can find it. Let's have a look. I just look for MPRO quickly, sorted by name.
Sort by date.
There you go. I've got it, the 17th one. So we can provide all sorts of data. So when you look at these downloads, I suppose I may as well talk about it as I'm talking about it. Whatever's in the directory called aligned is the data as it's presented in Fragalysis. The original crystallographic data are in the crystallographic folder. And then on the top level, we include all sorts of other data. So for example, we have a metadata file. And if I open that, you'll see that, you know, it's not ideal because we're not presenting it on the front end, but we can include additional information. So for example, we have information about the site names, which is included in Fragalysis. We have all of the smile strings. And then in this particular file, we've included the PDB code for any of those structures that have already been deposited to the PDB. And I think sorry, clicking on too many things. Also for MPRO, there's an Excel spreadsheet that contains more data about what's going on from a crystallographic point of view. So this is what the beamline guys use to track what's going on with compounds, and it also links stuff back to the moonshot as well. If it'll let me open it, I can have a look and tell you what's in there as we're working on MPRO anyway. So the answer is no, we can't include biological information on the front end of Fragalysis, but we can include it as part of the download package that exists. So you can see we can include pretty much anything we want, and details of the screening libraries we're using, how many soaks were done for each molecule, and things like the specific links on Fragalysis, although this is an old version. But yeah, are there any more questions? Chris, is there anything else, or
Okay.
shall I?
Yeah. Can you save the steps you've taken during a session so you can continue later?
Absolutely, yes. This also comes later in the tutorial, but I'm happy to go through it now.
But if you can save that for later if you want to.
I'll save
Okay.
it for in a bit. We'll get through the
Yeah.
science, and then I'll tell you
Yeah.
about saving steps as well.
Another question: can it be used to merge fragments?
So at the minute, there isn't an algorithm within Fragalysis for merging fragments. But what came out of the COVID moonshot work was a piece of software we developed called Fragment-based. I'll just flash that up quickly. So traditionally, how users have used Fragalysis is to look at the overlay visually of fragments and to sort of sketch down ideas, and then go further and search libraries elsewhere. But out of the COVID moonshot work, we had an algorithm come out that will take those initial hits that exist and work out where those overlays are in an automated way, and generate confirmations of those predicted mergers, and also do a bit of work to minimize within the protein structure to see how they might interact as well. So if you're interested in Fragment-based, you can have a look at it on GitHub. I guess I can copy this into the chat. And if you have any more questions about those sorts of methods, let me know. Of course, all of this work, eventually we hope to implement algorithms in the back end of Fragalysis and make them accessible to the user, but we're just not at that point yet.
There's another question about can you use it for peptide fragments?
Can you use it for peptide fragments? Yes, I don't see why not.
Yeah, you just need a peptide library.
Yeah, exactly. Yeah, I mean, as a screening platform, we don't offer a peptide library, but if you wanted to come along and screen one, then you'd be more than welcome to come with your own library.
Okay, that sounds great. Let's move on. That's all the questions up to date, I think.
Okay, awesome. So I talked about filtering these hits, so I'll go through very quickly. So when we upload the data, another thing we do for these fragments, now that we're treating them in isolation from each other, is we calculate some molecular descriptors. So we're just using RDKit on the back end to calculate these. And then in this modal, if you pop it up, you're presented with what the descriptors are, the minimum and maximum values for them, and also some extra buttons. So if I wanted to have a look at molecules in a certain range of log P, I could do that. So I've just filtered it down. So I'm looking at molecules with a log P between 2.64 and 4.3. And then if I also wanted to sort by that, I could move the priority of that up and down in the list with these up and down buttons. You can also change the order, so you're sorting by either increasing or decreasing log P. And if I switch them down, you'll see now that it's going in descending order instead of ascending order. We can use multiple filters at once as well. That's not a problem. So we can also filter towards higher molecular weight things if we wanted to see hits that are probably further along in the process than the original fragment hits. And we could move that up as well and change the priority of sorting there. You can look at your polar surface area if you wanted to as well and filter that down even more. So you can sort of start to think about things you're interested in looking at in the first place. I wouldn't say this is a good way of prioritizing your fragment hits in the first place, but it's sort of a tool that exists to help us. Actually, it's more useful in the computational side of things, and I'll demonstrate that in a little bit. So if you wanted to get rid of these filters as well, you can just click on the clear filter button and they should disappear, and you should go back to be sorting by site and the crystal number, which is the default sorting method. So in addition to that, what we have is a couple of predefined filters which will fill up over time once we learn how people are actually prioritizing these molecules. either in Fragalysis or elsewhere. So we've got, for example, a rule of 5 filter. It's not working properly, but if the values don't fit within the rule, they should be coloured in red here. And a rule of 3 filter as well. So yeah, and we can also turn that off. And we can, at the top here as well, clear the list. So we can turn off everything that was on in the central viewer. And we can do the same for sites as well. So if we had multiple sites turned on, for example, and we wanted to sort of get a fresh start, we could click on clear selection and that'll turn everything off. Okay, so enumerating new molecules. This is the interesting bit. This is where the graph network comes into play. So if we have a molecule that we're interested, let's just say for argument's sake it's this exact molecule, which is just the first one in the first site. I've not selected it for any particular reason. Maybe we turn on everything. In fact, you can do that with the A button. Except the surface. Let's turn on the surface as well. Okay, so you might look at this ligand and think, okay, well, it's quite well in this pocket here, but there's loads more of the active site pocket over here, and how am I going to access those areas of the protein as I'm developing my ligand? So one way of doing that is with this graph network enumeration method that we've spoken about. So that's those molecules that are available from enamine. And so the way I was talking about the graph network earlier, you might think, oh, well, how do I know how I can elaborate my molecule? And what's it going to look like? So in Fragalysis, what we've come up with is the concept of vectors on top of your original hit. So in this example, there are different elaborations represented. So an arrow represents a direction for elaboration, and a cylinder represents a change in the linker. And anything that's red, that means that the graph network is aware that there's a vector there and it could be elaborated upon, but it can't find any compounds to do that elaboration. Yellow means there's between 0 and 10 compounds available in that direction or that change, and green means there's more than 10 available. So if we then click on one of these vectors, we're presented with a list of molecules over here that are available in the graph network. And so you can start to look through those. And if you saw things that were interesting to you, you might want to categorize them. So you might want to say, buy this, I've decided I want it. And so you can just click on whichever molecules it is that you're interested in. Then to use a new filter, you can just click on enter on the filter and then it'll change color. I believe, does it work still? No. You used to also be able to use the number keys on your keypad to change that color, but I think that has now changed. Okay, so just click and enter. And you can add the categories as well. So for example, avoid. And once we have that list, what we can do is we can download a CSV file that contains the information on those selections. Hopefully. Yep. So what we're presented with when we do that download, I'll just close the one that's in the background so I don't confuse you, is a list of the smile strings of those new molecules you've selected. In the mol column is the smile string of the original molecule that it came from. So if we had a large selection over a number of different molecules, you'd be able to trace back to what the original molecule was. And then also this vector notation that we're talking about. So what this describes is the original molecule and where we're making the change as well. You're also given the classes that you just assigned those things to. So buy this and avoid were the ones I picked, and the class color that you picked. So you can refer back to your original page if you want to, and sort of correlate this data together. And so if you sent this column off, not that column, sorry, this column off to enamine, that's where you'd get your quote to buy new compounds from. So that's all well and good. We've got a list of molecules that we can buy. But, you know, how do we know that any of these is a good idea? So if I turn our surface back on again, and we go with the hypothesis, the very simple hypothesis for argument's sake, that we want to make a change in this direction to try and get further into that pocket or make more of a useful interaction over here. What you can do is you can click the Shift key on your keyboard and then click on any of these molecules, and that will do an estimation of the 3D structure of that ligand. So this estimation isn't a computationally intensive one, it's a ligand-based method. So a ligand-based method means using information about the ligand that we already have to estimate the new 3D structure. And how that works on the back end is it uses RDKit to generate 100 different conformations of the ligand that we're interested in. So going from the 2D SMILE string to 3D estimations of what it might look like in reality. It then aligns those new estimated structures to the reference structure. So this gray molecule in this case that we've put into the algorithm. And then for each of those aligned conformations, we use a score to decide which one is the best representation. So I'll show you quickly what that looks like. I was going to go through this in slides, but I think there's no need to. Okay, yes. So for the conformer generation, if you can see the slide, selecting the best one. So we use something called Sucos, which again is available on GitHub. I will copy and paste that link. I think I can do it to, I can just do it to organizers. Chris, if you could copy and paste, that would be great. Thank you. Yes, so it's something called a shape and color-based score. So the idea is it looks at the features, so the functionality of the original molecule and the new molecule, and sees how well they correspond. So you could think of that as like a pharmacophore alignment score. And so that's the first part of the score. The next part of the score is a shape-based metric. So it looks at how close the ligands are together in terms of their shape. And then this lambda value here is just some sort of weight that was optimized through the development of the algorithm until they got something they were happy with. What I've shown down here, maybe I'll zoom in on that a little bit, is why using this sort of score is better than using something like an RMSD. So in each picture, the cyan molecule represents the docked conformation, a docked conformation. It could also be generated in this method. It's just a different one that's been used for the figure. And then the purple molecule represents the conformation from an actual crystal structure. So if you look at the RMSD values for those, so this is all atom RMSD, this estimation of the structure over here is given us a 1.9 angstrom RMSD, and the one on the right hand side is given us a 3.1 angstrom RMSD. So you can get quite high RMSD values based on small deviations in small bits of the molecule. But when you compare that to a visual overlay, so if you actually looked at it physically instead of just relying on an RMSD value, what you'll see is quite different to what the numbers will tell you in a lot of cases. So the Sucos score was developed with the idea of features and shape, giving us a better overlay metric, and indeed it does seem to give us a better idea of what's going on. So with Sucos, a score is between 0 and 1, with 1 being perfect and 0 being nothing, and anything above 0.5 is better than random. So yeah, you can see this overlay here, which if you were visually inspecting it would say is awful, is getting a lower score than this one, but the RMSD for this one is worse. So we think it's a better way of doing it, and it does seem to generate quite quickly in the background conformations of molecules. So for something that you're doing on the fly in a web application, it's quite useful as a method. And you can go through and turn those off by just shift clicking again and turn them back on. You'll notice that because we're actually doing the algorithm in the background, if you turn on an estimated structure. So here's an example of one that we might be interested in because it's making an extension to the molecule and reaching back further into that pocket. But if you turned it off and then turned it on again, you'll get a different variation of that structure. They're usually quite well aligned with the original molecule, but just because of the way the algorithms implemented, it's generating 100 random structures, so the best one that comes out can be different every time. Okay, are there any more questions at this point, Chris?
Will XChem be able to visualize vector modifications on the compounds? I presume that means the user modifying the compounds.
I'm not 100% sure what that means. So will we be able to see what our users have done? Is that the...
So instead of just using this sort of library of compounds that are there, can the user edit the compound and see how it would align?
So at the minute, no. So so you mean adding custom compounds to this sort of functionality? Not yet, no. I will show you another feature in a while. So because we don't have the intensive algorithms on the back end, and this is sort of just a guess at what we could do to do this, we have a feature where you can import data, computational data, and visualize the structures, which is probably more robust anyway. But yes, it's a good idea for a feature. By the way, if anyone has any ideas, there's a submit idea button at the top of the page. Please go ahead and tell us, because we love it when our users tell us how to do things, because we have no clue. And you can also get me on Twitter as well if you want to.
So another question, do you estimate free energy binding at any point?
Again, not in the front-end application itself. I think that's something at the minute that you'd take it offline, run the calculations, and then reload the structures so you can look at them in the context of the original fragment screen.
Yeah, that sort of calculation usually takes a long while, so I don't think it's the sort of thing you'd want to do in a web application anyway.
Yeah.
Does the ligand enumeration take into account interligand clashes, steric or due to ionization?
No. Yeah, no is the answer.
Okay. That's it, we're up to date.
Okay, cool. Yeah, I guess there's just a couple more buttons down here to go through. So there's a clear selection button that will just clear everything and turn it off. And then there's a select all button. So if you were really interested in all of the molecules that went off one vector, then it might be an idea to just select them all. The 2D image up here is supposed to represent what vector you're on. I think I've recently messed up the graphics, so I apologize that it doesn't look perfect. When I get it fixed, that sort of faint green line that you can see there will be more obvious and will just represent in 2D where the elaboration vector is, so that you can keep an eye on that. There's also a little bit of a summary up here as well. So you can look at the number of molecules you've made in your selection from the graph network, the number of different vectors you've explored. So for example, if I've got those in my selection and I go to a new vector and add some more, it'll tally up the number of vectors you're looking at. It'll also give you an estimated cost, which I wouldn't trust. We call this the Anthony-Bradley algorithm. So Anthony was the first person that started working on Fragalysis, and I think we knew roughly what the cost of a molecule was. So the algorithm on the back end is £500 plus £250 for every other molecule. It might be close sometimes to the actual cost. I think, you know, one of the improvements we'd love to make going forward with Fragalysis is working more closely with those vendors to see if we can make a better estimation of the costs. And that is it on the vectors. So the next thing we can think about, before I move on to the computational data, somebody asked about sharing the state of the page. So there are two concepts in Fragalysis for this. I'll show you the first one. So I imagine most of the people on the webinar won't have a login for Fragalysis because it's done through Diamond's login system. I can show you in a little while how to create one of those logins if you want to, so that you can use the other feature. But the most simple idea in Fragalysis is the idea of a snapshot. So it's either you want to save the state of the page as you are for your own work, or you want to share what you've done with a collaborator. So how you would do that in Fragalysis, you can create a static snapshot of the page by clicking on the share button. So if I click on share and it decides to work, yep, there we go. So we have, this is just a randomly generated stamp. I'll explain to you in a minute why that happens. But what we get is a link that should recreate the state of the page as it was. And you can open it in the browser and there you go. We can see exactly between the pages what those were. I suppose I could also copy and paste that into the chat so you can all see the same thing as well if you want to. And the other feature is called a project. So I'm going to navigate back to the main page and log in now, or from the sidebar. So this is just...
So just a quick question on that link you shared. Is that
yes.
permanent?
It's supposed to be permanent. So the problem with this sort of thing, especially with Fragalysis and the Moonshot project specifically, is that the data changes very quickly. So if this, the question is, are you trying to capture the state of the data as it was at that point when you saved it? Or are you also trying to account for changes to the data that have happened? So we still haven't quite wrapped our head around that question. So at the minute, the snapshot is a snapshot of a specific state of the data, and we haven't quite worked out how to maintain that over time. So when we update the data, sometimes the snapshots break a little bit.
Yeah, I was just thinking that if somebody wanted to use it in a publication or something like that.
Yeah, I think that problem has already happened, much to my sadness. I think there's a project link in a publication somewhere and it doesn't work, which is not ideal. I think, yeah, I can go through another feature of Fragalysis as well that might be more helpful. So the snapshots are great if you've done a lot of work in Fragalysis, but what we've found through the Moonshot is actually what people want to do most of the time is just say, look at this ligand. And so I can show you, for example, on the COVID Moonshot pages, if we go there, if we go to structures on the COVID Moonshot page, another copy and paste, I think, then you're presented with all of the different ligands that have come in through MPro, and there's a link for each of those 2D images that says view on Fragalysis. And if we click on one of those links, it's using something called a direct link now. So these links work in a way that they're focusing on a specific molecule, for example. So here we're saying turn on the molecule that starts in X0161, turn on the ligand, turn on the protein, and turn on the contacts as well. So if you're worried about the data changing, but really what you're sharing is the ligands, you can access that through those example links.
That's it for questions. We're up to date.
Okay, awesome. Where was I? I was going to log in. Have I logged in? No. Okay, so I'm going to log in. I was about to log in. Let's close these. So if I log in, so this is Diamond's authentication system, and you can register for a Diamond account if you would like. So if you click on the login button on Fragalysis, you'll come to this page and you can register if you like. I think, usually this is used for our user program, but you can sign up anyway without being a Diamond user. So once you log in, it should load a page. Excellent. So let's just very quickly turn some stuff on that looks a bit different from default. So let's say I have started with a ligand and I've turned on a molecule I'm interested in. And that's the first save point I want in my project. This is the first ligand I'm looking at, so I'm going to create a snapshot of that and save it. So if you click on save, you can start a new project. So I might just call this MPro demo. You can give it a description if you like, so demonstrating projects. And you can also add some tags to your project as well, so things that will help you to remember what the project's about. So I guess demo is a tag for this one. And then you can create the project. So when you create the project, that initiates the project in the back end. And a project is essentially a collection of snapshots. So you can then give a title to a specific snapshot. So you might say, for example, enumeration of aminopyridine like molecules. And the description is auto-generated, but you might just want to put some more details in there, like I clicked a vector and found something I liked. And then we can save that snapshot. And then what will hopefully pop up, we obviously need to optimize this so it doesn't reload the page when you do it. But what pops up down on the bottom right-hand side is a project history. So this is the first node in our project, and if you hover over, you can see what the title of that snapshot you just gave was. So then let's say I turned all of this stuff off, not interested in this anymore. I'll look at a different site, different type of molecule. That's a molecule that has 0 vectors, which is useful when you're trying to do a demo, but okay. We'll stick with that one. We can make an additional save point, different enumeration, nothing available, and save that. And then in our project history, we've got the different steps that we've made and the different snapshots that we've got. And if you pop that up, you just get a little bit more detail. So you get the descriptions that you've saved and the date and time that you did them as well. And it indicates which snapshot you're currently on too. And if you click that little link button, it'll open the link so that you can copy it and send it to someone to look at. So the projects themselves will usually be specific to a user, so you don't need to worry about other people being able to see your projects at the minute. It's sort of a user by user thing. Okay. Another interesting thing you can look at as well is the timeline up here. That works well. So if I don't know, refresh the page and do some new stuff, let's see if this works. It's because I'm on a project. So let's go from the beginning on a, where am I? MPro. Turn a few things on.
Quick question while you're loading all that lot. Say the result set contains 1,000 compounds. Is there any algorithm which recommends which are good things to do?
That's a very good question. I wouldn't have a job if there was an answer to that. So I think there are, you know, not in Fragalysis is the quick answer, and it's a problem we're really thinking about. But I guess maybe what the question really is there a way to suggest molecules the way that Amazon suggests that you buy a Hoover after you've just bought a Hoover, right?
Yes, Yeah, or notices that you're browsing through Hoovers and says, why don't you choose this one?
Yeah, exactly. I think, you know, that's a fantastic idea. I don't know whether others have implemented something similar before. I think it's definitely an opportunity as well, right? It's something that's going to require a bit of machine learning and using the data we already have, and maybe thinking about how people make decisions already. I don't know how easy that sort of thing is to encode. I mean, you've got to have, it depends how complicated you want to be.
Yeah, I mean, I think you're right. It's a fantastic opportunity for somebody. So
Yeah, absolutely.
yeah, I'm sure there's lots of AI specialists who are watching this who are madly scribbling down things to do.
Because they can see that we can't do everything yet.
Yeah.
We're just scratching the surface.
Okay, that's it for questions.
Okay, fantastic. So I was just going through this actions list, which we've not done much with yet, but it just sort of gives you an indication of the actions that you've done in the interface. So you can look through and work out how you got to the point you're at. Okay, so we can close that. So the final bit that I've mentioned and would like to talk through, I'll just clear all this, is the idea of uploading new designs. So a lot of the time, I think there'll be people in the audience that are familiar with this and maybe others that aren't. And we've touched on it briefly already. When you're designing new molecules after that enumeration, when you're trying to work out which things to buy, you need to know what they're going to look like in 3D, whether you're making any new interactions that might be useful, making sure you're conserving things that you already know from the experimental data. And what that comes down to really is a lot of hardcore algorithms. So there are different algorithms. You might want to do some simple docking, you might want to do some MD simulations. Whatever it is, the main thing you're looking for out of that is a 3D structure, and also some sort of indication of score or a metric that you can quantitatively use to help you narrow down your selection. And so while we know that the algorithms are really important, we haven't focused on putting them into Fragalysis yet because it's a big piece of development work. What we have focused on is being able to get the results of those into Fragalysis. So if you click on the drop-down arrow on the right-hand side, for the moonshot, for example, you'll see a bunch of different data sets with very cryptic titles. I apologize. But if you click on one of those links and then click on the tab, you'll be presented with a list of molecules. So these molecules, the Tim-Dudgeon Feature-Stein molecules, are molecules that have been designed from the fragment network, but in a much more high throughput way, and then they've also been docked and scored as well. I will send a link shortly. That, in fact, don't need to send a link. If you click on the link to dataset button, that's just in that menu bar, you will be sent to a page that has nothing on it. Let's try a different one. Should have tried that beforehand, shouldn't I?
Yeah,
okay.
It's the wonders of live demos.
Yeah, it's fantastic. Okay, so usually when you click on one of those link buttons, you'll be directed to a page that describes the method that's been used to generate the data set. So that's sort of the quickest way that we can show what the actual science going on behind this data is. And then so for these data sets, when you upload them, I can show you the upload page shortly. You essentially have a 3D structure, so you can turn on a ligand and it'll present them as a licorice model and show you where it is in reference to the reference protein and the aligned structures. You can also do very similar things to, when it works, you can do very similar things to what you can do on the left-hand side. So usually you'd be able to turn on side chains and interactions as well, and the surface. And I think so what this is supposed to do is show you what the inspirations from the left-hand side are, so you can turn them on. and view them in the context of the initial hit. So maybe what I'll do is upload a new set and show you how that works, and hopefully it will update and all the features will work. So I will go to a new tab. So I'll copy and paste this quickly. For anyone that wants to contribute any 3D designs to the COVID moonshot, I'm sure you're all dying to. If we go to this page, we're presented with an upload page. So the first thing we need to do is say which target we're uploading designs for. What is very important is the SDF file. So because of the data that's in Fragalysis, there's a specific specification, which I'll go through shortly, which is currently living on the Postera forum. So that's a forum that's part of the COVID moonshot, and it's there because this feature was developed during the COVID moonshot. So we need to choose a file. I believe I put one on my desktop somewhere. Yeah. Okay. Then we need, if you want to provide your own PDB files, you can as well in reference to those ligands. And then you can either validate that your files are compatible, or you can upload them directly. You don't need an upload key. Actually, we've turned this feature off for now, so just ignore that. And then if you're updating a set that already exists, you can select that set, and it should play ball. So after we've uploaded our SDF file and given a target and decided to upload it, we can submit, and that kicks off a job in the background. So it's sort of like dynamically processing all of that data and putting it into the database. Now, sometimes it takes a bit of time. So this is telling me I just need to update the set instead of submitting it as a new one, because there's already a compound set that has that name, which is fine. We can go back and do that. So if there's any problems with your files as they upload, what just happened to me will happen to you. It'll tell you. So I believe that was a compound set that's now just disappeared from the database somehow. There's magic going on behind the scenes, I'm telling you. It's a mystery to me. So let's find the file, and I will quickly change something that we can do so that we can do this live. I'll just open it in TextEdit. And it wanted me to change the method name. So did it want me to change the method name? Let's try it. Test upload. We will call it. That's fine. So I can show you very briefly, I suppose, what this SDF file looks like. So because we want to include a lot of data in Fragalysis, like scores, and also some context to that data, the only difference from a normal SDF file with one that you would upload to Fragalysis is that we have to define a few things. So we have to provide a description for the scores that we want to add to our molecules in the property fields, and we need to give a URL for the method, and then just some extra information about who's submitting it, when it was generated, and what method they used to produce the data. So I'll save that and then give it another go. And we might have more luck this time, or we might not. Are there any more questions while this upload's happening, Chris?
No, but I noticed the file you're uploading is a 2D file, is that correct?
No, it's 3D. So that first, I can go through it. So the first molecule is just a blank molecule. So I think this has just been generated in RDKit by the person that's generated the file, and so it's just that initial molecule. But then if you go down and look at the rest of the molecules, they'll all be 3D structures with those values for the scores that we've defined in the reference molecule at the top.
Yeah.
This might take a while. Oh, there we go, it's uploaded. So when something uploads successfully, it also generates links so that people can download these things from the back end, and you can check them yourself and make sure that everything's uploaded if you want, and access the proteins that you specified in the file. So now if I refresh the page, I think we need to reload it to access the data. And it should be called something test upload. Yep, there we go. So that's the upload we've just done. We can go there and see those molecules we've just uploaded, and now it works. So now you can click on the F button, and that in the SDF file you upload, you can specify which initial hits inspired your new design. And then so in Fragalysis, you're allowing the users to see what's inspired you. So if we then turn some of these on, or even all of them, if I click this button at the top, it'll turn on all of those inspiration ligands, and we can look at the original hits in the context of the new design and start to see where it came from, and how well we're conserving those original hit positions as well. And not only do they turn on here, it's reflected in the left hand side as well. So if we turn on the sites that are relevant, site 8 and site 3, for example, we should see, maybe if I sorted by site, it would be useful. Let's go by site. So we should now have, if it's worked, site 8 at the top. So you can see, for example, this one is turned on the left hand side as well as the right hand side. We also have the functionality of filtering, as we do on the left hand side as well. So these values here in the columns on each of these molecule cards refer to scores. If you hover over them, you'll get the description that was defined in that uploaded SDF file, so you know what you're looking at. And if you click on the filters, you can start to use those different scores to try and narrow that list down a bit. So if we wanted to use a particular score, and positive is better, and the bigger values are better as well. Which score am I looking at? GBSA and what 0? So this score here, for example, at the minute it's going up. So let's sort it in the opposite order. If that was a golden bullet score and the big values were the best ones, and we could sort by them and start to look at them. So we might do that sorting and filtering and then start looking at the actual visualization. So I turn all of them off and all of them off. Let's not turn all of them off. Just get them off. Maybe clear that side. It's failing to be responsive, which is excellent. Love it when it happens. Just give me a second, I'll reload and get back to where we were. Are there any more questions while I quickly reload this?
No, there's no questions, but I have a question.
Yeah.
If somebody has docked these in and used a flexible docking tool, then
Yes.
sidechains may have moved and things like this. Presumably then it might not look very attractive in your Fragalysis.
It might not do, no. So I think, through most of this demonstration, we've focused on having this reference protein. And probably at that point, you'd want to turn on the actual protein structure, right? So you can upload, when you upload the SDF file, a zip file of PDB files as well. And if the PDB file is one that you're uploading, you just specify that in the SDF block.
Okay, that sounds good. Yeah, and then that would solve that problem.
Yeah, so you can look at those differences. Okay, so I was here, and I was going to do something. I don't remember what it was.
I think you're just going to show the interactions with the novel ligands.
Yeah, probably. So, oh, I know what I was going to do. So we'd done some filtering and sorting and decided, and then I was going to go through the scenario of if you'd had a look at some of these visually and, you know, made a decision on what you wanted, then you can make a selection of ligands, and they'll appear in a summary in the selected compounds tab. Another feature that exists over here to try and help you navigate quickly through the ligands when you're looking at them. So if you had a view of, you know, different inspiration molecules that you were looking at and you were comparing, for example, so let's turn some on, and you wanted to look at other molecules that might be interacting in a similar place, you can just click on these down arrows and it'll very quickly navigate through all of the ligands for you, applying the same settings. So for example, if you had the side chains turned on for one of these and you click the down arrow, it'll turn the side chains on for the next molecule, and so on. So you can just apply the same settings to the next molecule in the list while you're quickly navigating through.
Okay, there's a quick question here. What criteria do you use to choose the reference structure?
Good question. So when we are aligning the things before they go into Fragalysis, we use the structure with the longest chain and the best resolution with the most correspondence to the most other structures. In Fragalysis itself, I believe it is randomly selected, which is a bit rubbish. We should probably use the same reference that we use to align to.
Yeah. In your display, can you show B factors? Not
yet, no.
Okay.
Fragalysis is still very much under active development. So, you know, it's a proof of concept and an idea for what we can do, but we're very, very much open to working with other people and getting their ideas. So, you know, the good thing about this for me is that it's getting the concept out there for other people. And if you guys want to feed back to us, please do, because, you know, things that you think of as chemists, we won't necessarily think of as crystallographers and computational chemists as well sometimes.
You're up to date with questions.
Okay, sorry, I just needed a bit of a cough. I apologize. So that is essentially everything in the interface so far. There are a couple of extra buttons as well. Like I said, there's a report issue button. I'll just show you how to use that quickly, just in case any of you ever need to. So it'll take a screenshot of your application page. So if you go to application window and click on that, or if you go to the Chrome tab and select the one you're on and give it permission to share, then what it'll do is it'll take a snapshot. And so if you had something like, I don't know, a ligand button didn't turn on, you could circle it in red. So we could say whatever your name is, your email address. I'm going to just put ignore on this and then put a description in like ligand isn't working. And then you could submit that idea and it should, if all is good, it should actually say report issue, not submit an idea. Yeah, so that'll pop an issue on our GitHub page. So GitHub is where we keep the code and where we develop it. So once you've submitted that, you have a link and you can keep that as a reference. And we can also contact you for more information if we need to, and look at the state of the page when you captured that snapshot and see the image. And the ideas thing works in exactly the same way. So if you had, for example, like a slide or something articulating your idea and you wanted to include that in your image, then when that pop-up comes up to share something, just share that application slide instead and it'll be uploaded. Yeah, we've mentioned the moonshot a few times, so I suppose I should go through a quick example of how this was used in the COVID moonshot. And I'll extremely quickly at the end point out where the Python API is for people who are interested. I think I zoomed in far too much on this. So yes, in the COVID moonshot project, there is a molecule that was designed in Fragalysis in this concept of looking at the overlay of ligands together and seeing how you might merge them together. So these are snapshots from Fragalysis. There are 5 different ligands that were used to design a new one. And I guess we could turn them on, and I'll demonstrate another feature of Fragalysis to you while I'm there, which is that it's really terrible on a tiny screen. No, that was a joke. So if I just get these sites on quickly, and then I'll search for the inspirations, and I'll show you what it looks like live. So now I've got all my sites on, we can look at these different molecules that are the inspirations. So if we look for Mpro X107A, for example, I might just search 107A, and up pops Mpro 107A. I'm just going to clear everything first. Let's start from scratch. Too much computational stuff going on. Okay. I suppose this is how live demos go. And we really need to turn on all sites button as well, is what I've just realized. 107A. So let's turn, that was on anyway. Fantastic. So we've got 107A on. Then we can clear the search, turn on 678A. Which one is it? That one, 06788. Then 1382, for example, 434, and 995. This one. Okay, so if we bring that up bigger, I'll show you what I did to look at these overlays with each other. If you zoom in a bit. So I had to sort of reproduce this process recently for a manuscript I'm writing. So what I did was I changed these representations. I just thought it was a nice way to look at it. So I changed them all to licorice so that we can look at them more easily together. And then you can sort of see, I'll turn on a surface as well. Why not? So these are all crystallized in the active site, and they're crystallized in parts of the active site known as P1 and P2. So that's pocket 1 and pocket 2 out of the 4 different pockets where the native ligand sits. And you can see quite clearly from these ligands opportunities for elaboration, I think. We just turn that surface off again quickly. So you can sort of see how the moonshot contributors visualized those hits together and designed a new molecule. And if we look at that molecule back on the slides, we can see that it's a combination of all of those molecules quite easily. And so this molecule was selected by the moonshot guys because it was a merger to start with. So it was really clear that they'd used the experimental evidence in front of them to design it. And because it is synthetically accessible. So there's an algorithm that was used in the moonshot to see how easy things were going to be to synthesize along with enamine. And it's, you know, it's a really nice story about the success of Fragalysis, because we started with that submitted design and we had a bit of inhibition, and we've slowly, with some med chemists, made iterations of the molecule, and we've gotten better and better IC50 values, and that thing's now gone into in vitro assays, I think, which is really nice. Okay, the Fragalysis API, I will very, very quickly find it for you and just talk a little bit about it. I won't demonstrate it. So all of our codes on...
There's a quick question while you're doing that. Can we run Fragalysis on a local server?
If you wanted to, yes, you could. So the Fragalysis code bases,
I think you showed the GitHub.
Yeah. So this is for the API. So the Python API is a slightly different thing.
Yeah.
The Python API is for accessing the methods in Fragalysis and the data in Fragalysis that is live for Diamond. If you wanted to deploy your own version of Fragalysis, that's also possible. If I find one of the documentation pages, it's all containerized and it's deployable on normal cloud infrastructure. So I can point you to that. It goes into a lot more detail about the ecosystem and how it's deployed and stuff as well, which I think is a different conversation altogether. But if you're interested, that's a good starting point for you. And also just reach out to us and we'll go through it with you in more detail. I'll copy and paste my email address and Twitter handle in a minute. I think we've got 5 minutes left. So I just wanted to extremely quickly touch on the Fragalysis API. So it serves a few functions. Actually, one of the most important functions it serves is preparing the data to go into Fragalysis. So that process of taking all of those bound state structures and pulling them apart into the ligand and the protein. And I think, that's probably, and also aligning them all to the same reference frame as well. I think that's probably quite useful for people that are working in computational chemistry as well. So if you want to do that, I would recommend looking at the API, which I'll also copy and paste, and looking at the documentation and seeing how you can do it. There's also some example notebooks in some of these different areas on the API where you can see what you can do to access different bits of data and query our databases, and also to use the graph network for your own purposes. So there's a really nice thing in the Python API where you can put in a smile string and it'll spit back a data frame for you, so a table with all of the elaborations that are available from the graph network. So if you have something, if you've not uploaded data to Fragalysis, you can still make use of that resource.
And I presume people can contribute towards this as well.
Absolutely, yeah. I mean, we don't have any contribution guidelines up there yet. But if anyone is interested in working with us, just reach out or make pull requests, I guess. That's absolutely fine. Okay. If there are no more questions, I think I'm done unless there's anything else you'd like me to show.
No, I think you've done a fabulous job. All the questions are answered and you've got almost perfect timing as well. So I don't think we could ask for any more.
Okay, ideal.
Yeah. So I guess there are lots of people who want to contribute to the COVID moonshot. So this gives them a great point to as a starting point to contribute. I think it's a fabulous initiative. And I think you've given us a brilliant insight into how to do this.
Excellent. I'm glad you enjoyed it, Chris.
Yeah, well, many thanks. And as I said to the others, if there was a virtual round of applause, I'd be pressing the button now.
Thank you. It's always hard to gauge the audience reaction when you can't see them or hear them.
Yeah, I think there's been many compliments on the presentation. You've given an excellent workshop.
Okay, thank you very much, everyone.
Thank you, and I'll be closing down the webinar now. Thank you. Bye-bye.
Thanks. Bye.